So let's start with Neisseria gonorrhoeae. So when we're starting with Neisseria gonorrhoeae, so first let's have a look at morphology. So now, so you take now suppose a patient is come to you, okay, and there is the patient is complaining of urethral discharge, and you are thinking that it can be a case of gonorrhea. So you take that urethral discharge and make smears from that. In that smear, you will observe that the gonorrhea, that is the gonococci, they will be appearing as diplococcus. Okay, then the adjacent sides of the organism is concave and the shape is typically kidney shaped, and you will find it within the polymorphs. That's inside the polymorphs, you're going to find them, and you will see that the numbers of cocci are in hundreds. Then also you're going to find pili on the surface. What are pili? So pili are actually hair-like structures, okay, or which are several micrometers long. You will see that they are usually used to uh, attach the cocci to the mucosal surfaces, okay, any mucosal surface adhesion, and also they are going to inhibit phagocytosis. And the gonococci which have pili, so that's why they are called pileated gonococci, they are going to agglutinate human red blood cells. They're going to agglutinate only human red blood cells, but they won't agglutinate the red blood cells of other mammals. Okay, so this is the morphology. Now let's go to the cultural characteristics. So in cultural characteristics, see, Neisseria meningitis are comparatively easier to grow, but gonococci are a bit difficult. So you see that they're aerobic, but sometimes they can grow anaerobically also. Now, which is the best pH for their growth? It is 7.2 to 7.6, and the optimum temperature is 35 to 36 degrees Celsius. Also, essentially, you have to provide them around 5 to 10% of CO2. And the mediums or the culture media, you can culture them. Take chocolate agar or Mueller Hinton agar. Now, let's see which is the selective media for Neisseria gonorrhoeae. That is Thea-Martin medium. In a Thea-Martin medium, you should also have vancomycin, colistin, and nystatin. So, why do we say that Thea-Martin medium is a selective media for Neisseria gonorrhoeae? It's because they are going to inhibit most of the contaminants. The contaminants will be non-pathogenic Neisseria, meaning the Neisseria species, which won't be pathogenic, and also you can use it to inoculate urethral and endocervical swabs. Now, so if you see the colonies, now this was the till now what we saw was the optimum conditions and which media you use, but how the colonies look, we'll see now. So colonies are small, they're round, okay, they're translucent, they're convex, and they have a finely granular surface. So, see, in Neisseria meningitis, we saw that they had a glistening and a smooth surface, but this has a finely granular surface and has lobate margins. And the colonies are very soft, they're easily emulsifiable, means you can emulsify them easily. Now we can actually go to the biochemical reactions. In biochemical reactions, if you compare it with Neisseria meningitis, almost the biochemical reactions are same. Like the oxidase test, which gives a positive oxidase test. If you add oxidase reagent, like 1% solution of the oxidase reagent, you will get purple colors, deep purple colonies. So gonococci resembles meningococci, except it's going to acidify only glucose and not maltose in serum sugars. But you will see that meningococci is going to acidify both. Then, then we'll come to the a test called rapid carbohydrate utilization test. which is RCUT. It's a rapid sugar fermentation test. Meaning in this you're going to rapid sugar fermentation test. So mainly it's going to be based on the enzymes which are pre-formed in the bacteria. It's not going to depend on the growth of the bacteria and the sugar media. So that's why it becomes more sensitive and it becomes more specific also. Let's have a look at the antigenic properties. So antigenic properties, first thing you have to speak in antigenic properties, you have to speak about pili. Then you speak about the trilamina outer membrane. Then you can speak about proteins 1 and 3. Then you can speak about um, the outer membrane which contains endotoxins. Also, you can speak about other proteins which have roles in pathogenicity. So let's speak about antigenic properties one by one. So gonococci are antigenically heterogeneous, means what they do is they'll be changing their surface structures in vitro. Always they're undergoing changes in with respect to antigenicity. So what are pili? So surface structures which are responsible for giving them antigenicity are pili. So pili are hair-like structures. Their main function is what? They will allow the bacteria, that's the gonococci, to get attached to the host cells. So they act as virulence factors. How? See, they're going to promote attachment to the host cells. So main thing for a bacteria is attachment to the host cells. So pili will promote that. Along with that, it's also going to inhibit phagocytosis. So it will act as virulence factors by promoting these two things. Okay. Then also, you should know that they will be undergoing antigenic and phase variations. Then the second thing, which is a surface structure and responsible for antigenicity, is trilamina outer membrane. Trilamina outer membrane, and this outer membrane, which is trilamina, it contains many different proteins. Now let's speak about these proteins. So you have different types of proteins. There is protein 1, there is protein 2, there is protein 3. So, in protein 1 is a major constituent and it shows antigenic diversity, which helps in typing gonococcal strains. So, in every different type of a gonococcal strain, the protein 1 will have different diversity. It will have an antigenic diversity. It will be different in different gonococcal strains. Suppose there is one gonococcal strain, for that particular strain, the protein 1 is going to be antigenically constant, means it's going to be of the same type. Okay, it's going to show considerable heterogeneity among different strains. Among different strains, protein 1 is different. Now there are two types of protein 1s, 1A and 1B. Now in one strain, it will carry either 1A or 1B, not both. Okay, now comes protein 1 and 3. Protein 1 and 3, they'll be acting as ligands, okay, and help in attaching the coccus to the host cells. So first thing, protein 1 and 3 help in attaching the coccus to the host cells. Along with that, they also form transmembrane channels. There is porins, okay, porins. These are water channels, or they form transmembrane channels, and these transmembrane channels play a role in the exchange of molecules across the outer membrane. So protein 1 and 3 has two functions. First of all, helps in attaching the coccus to the host cells. Along with that, it also forms transmembrane channels, which helps in exchange of molecules. So protein 1 and 3 is done. Now we'll come to protein 2. So protein 2 is responsible for giving opacity to colonies. Now protein 2 is also known as the OPA protein, which is opacity associated outer membrane protein. Suppose there is a strain which has protein 2, that is the OPA protein. So they will form opaque colonies, but those which don't have this OPA protein, they will form transparent colonies. So first thing, OPA is responsible for forming opaque colonies. Along with forming opaque colonies, it's also responsible for attachment of the coccus to the host cell, and also it is responsible for the clumping of cocci. So you see in the urethral exudate smears, there are clumping of cocci. So OPA is responsible for this. So OPA is responsible for first, opaque colonies. Second, attachment to the host cells. Third, clumping of cocci. Now, The outer membrane also has some endotoxins. So these endotoxins are responsible for the toxicity in gonococcal infections. You have the gonococcal infections, they will be causing some problems to the patient. So this is because of the endotoxins. These endotoxins are lipo-oligosaccharides. Now also there is one more protein which is there are many other proteins which are responsible for pathogenicity. So there is another there is one enzyme, that is IgA1 protease. It's going to split also and inactivate IgA. So IgA is an antibody. Now let's see the resistance. So gonococcus is delicate. It is susceptible to heat, drying, antiseptics. So we will go into details of resistance point by point. Now you take the organism outside the body. It will die in 1 to 2 hours. You take an exudate outside the body, and then you are going to check it after 1 or 2 hours. It will be dead. Second thing, you have made a culture. The coccus is going to die in 3 to 4 days. But you make a slant culture at around 35 degrees Celsius, and but you have to keep it in sterile paraffin oil. Okay, it is going to survive. Now you want to, preserve it for years. What you will do? Take the culture quickly, freeze it, and store it at 70 degrees Celsius. Also, responsible for resistance is the plasmids. So there are several plasmids which have unknown functions. Okay, but these will be providing resistance maybe. Also, there are other plasmids. These are transmissible plasmids. These code for beta lactamase, which is an enzyme, and it causes resistance to penicillin. Regarding pathogenicity, how come, what is its pathogenesis and how? So you see that gonorrhea is a venereal disease, a sexually transmitted disease. How is the spread? The spread will be through sexual contact. Okay, so the spread will be through sexual contact, if you see. So the first step, the infection first step is gonococci. Whenever it comes in contact, it just gets attached to the mucosal surface or urethra. And who is responsible for this adhesion? It is pili. Now this adhesion is what type? First thing is pili is involved in this adhesion. And what is this adhesion type? This adhesion is rapid and firm. Okay, so this adhesion is rapid and firm, so even after micturition there will be no protection against the infection. And now the cocci have, they have attached to the mucosal surface. Now what they'll do, there are these intercellular spaces. Through those intercellular spaces they are going to penetrate means go inside, go deep. Okay, they will penetrate through the intercellular spaces. After penetrating, they will reach the subepithelial connective tissue, and this they will reach the subepithelial connective tissue when? Till the third day of infection. And what is the incubation period? That is to show the symptoms, there is one incubation period which is 2 to 8 days. After 2 to 8 days you get the symptoms. So let's see in detail how the disease is present in men. So first, start of the disease in men, it's going to start as acute urethritis. Okay, acute urethritis. There will be a mucopurulent discharge. There will be pus in that. Also, there will be gonococci in large numbers. So, first begins as acute urethritis. After beginning as acute urethritis, it extends to the infection will extend along the urethra, go to the prostate, go to the seminal vesicles, and reaches the epididymis. Okay, then the infection can also spread to periurethral tissues, causing abscesses and multiple discharging sinuses called Watercan perineum. Now let's see in women. In women, the initial infection obviously is going to involve the urethra and the cervix uteri. Now, see, very important is that the vaginal mucosa. is not affected in adults. Why? See, vagina has a stratified squamous epithelium. It is resistant to infection by cocci, and also because of the acidic pH of vaginal secretions. So, see, vaginal secretions pH is acidic, so that's going to provide some extra protection. Along with that, stratified squamous epithelium present in adults, women, okay, it is going to be very resistant. Now, the infection, it's involves the urethra, cervix, uteri, initial infection. Then the infection will extend to Bartholin's glands, okay, endometrium, fallopian tubes. Now, it can also progress as pelvic inflammatory disease (PID) and salpingitis can lead to sterility. And rarely you will find peritonitis with perihepatic inflammation, that is called as Fitz-Hugh-Curtis syndrome. So clinical disease is less severe in women. Okay. An asymptomatic carriage of gonococca is rare in men, but you're going to find asymptomatic carriage in women. So these are the other forms of illnesses. So there is DGI, proctitis, then conjunctivitis, blood invasion, because from the primary site of infection, it can lead to metastatic lesions. Then ophthalmia neonatal, it's a non-venereal infection in the newborn. The newborn will acquire it during passage through the birth canal. Then you come to the gonococcal bacteremia, skin lesions, hemorrhagic papules, pustules on hands, forearm, feet, legs. So this is basically skin lesions.